Last lecture, we considered differences between the blastula of the various animal models, including the rather symmetrical form of the yolk-pore sea urchin blastula and its indistinct animal and vegetal poles, pictured here, to the pronounced difference in the animal and vegetal poles and the moderately yolk-rich frog blastula, pictured down here. We also discussed gastrulation, which is where we see the formation of the three tissue germ layers, the ectoderm, the mesoderm, and the endoderm. And we also looked at the initial phases of organogenesis in vertebrates, notochord development, and neurulation. Now, in order to get a big picture view of the process, let's take a look at the overall process of human development. In humans, the newly formed zygote undergoes its first cleavage about 30 hours after fertilization. It becomes a blastocyst by the time the embryo reaches the uterus, which is around 6 to 7 days after fertilization. At this point, the trophoblast cells digest their way into the endometrium, and the chorion and the endometrium engage to form the placenta. Gastrulation occurs in the second week, neurulation occurs in the third week, and the bulk of organogenesis begins by the fourth week. By the second month, all major organs are established, and growth and organ maturation continues, including the brain, which continues to develop and produce neurons for months after birth. And now let's compare the extraembryonic membranes in two amniote models, the chick and the mammal. So the amnion is the innermost membrane that encloses the embryo of a mammal, bird, or reptile. The chorion is the outer membrane that surrounds the embryo, and in mammals, it contributes to the formation of the placenta. In reptiles and birds, the yolk sac has a function associated with nutrition, whereas in mammals, the yolk sac acts as a source of primordial germ cells and blood cells. And in the chick model pictured here, the allantois attaches to the chorion, which is the outer membrane, and allows for gas exchange, whereas in mammals, it becomes part of the umbilical cord. Now, today we're going to develop an appreciation for the role of cell division in the process of embryonic development, and we're going to develop a better understanding of the role of cell differentiation. And we're going to look at the pattern formation that takes place in Drosophila, aka the fruit fly. Now, today we're going to be examining development via processes that take place. Today we're going to look at three processes in particular that are involved in animal development. And those are cell division, differentiation, and pattern formation. And we're going to go ahead and start with cell division. After fertilization, the zygote begins to divide. And this division involves a modified cell cycle that allows for rapid division and no cell growth. This results in an increasing number of smaller and smaller cells that we call blastomeres. And this cluster of cells does not increase in overall size during the process. Now, this is a very organized process, and each of those early cells has a specific cell lineage that they will contribute to. A simple illustration of that is seen here. Through the application of a fluorescent dye, a map of cell lineage in the development of a slipper limpet, Crepugula fornicata, can be observed. When researchers inject the dye in a specific cell during early cleavage and then check to see where the dye ends up as development progresses, the pattern of distribution of the dye is the same across individuals of the species, indicating that the fate of a given blastomere during cleavage is already mapped out. Now, next, we're going to talk about differentiation. And differentiation is the process of progressive change that an unspecialized cell goes through that results in a cell with a specialized form or function. So, like I just mentioned, a differentiated cell is a cell that has a specific form or function. And they can be distinguished from one another by looking at the proteins they synthesize, by observing the specific functions they carry out, or by their morphology. And by that, I mean their shape and their arrangement, which is what you were learning about in the animal tissue lab. but the molecular decision to become a given type of cell is made before the changes actually take place. As soon as the molecular decision happens in a cell, we refer to it as a determined cell. And this is not typically visible, so we can do experiments by moving cells around to find out if the cell is determined or not. So let's say we select a donor that's in early development and its cells have not become determined yet. If we transfer tail cells to the head of a recipient shown here, what's going to take place is those tail cells will develop into head cells in the head. However, if we select a donor later in its development where determination has already taken place and we transfer the tail cells to the head of a recipient, what will happen is those tail cells will develop into tail cells in the head of the recipient. And now we're going to move on to pattern formation. Pattern formation is where we see changes in symmetry from a radially symmetrical zygote to one that shows polarity, features such as an anterior posterior axis or a dorsal and ventral axis. Now, embryogenesis in Drosophila, the common fruit fly, is considered development from a fertilized egg to a larva. The larva's primary role is to eat. And then after metamorphosis, in which the larva develops into the adult fruit fly, its job is to breed. And when you look at the peer-reviewed literature, you will find quite a bit of information about this tiny little fruit fly. Of course, much of that being the fact that they're very easy to take care of. They don't eat a whole lot. They have a lot of offspring, and they don't live very long. So they're ideal models for genetic studies. Now, the development planning of Drosophila begins before fertilization, when specialized nurse cells transfer their mRNA to the oocyte or developing egg. These transcripts are maternal in that they are from the egg-producing organism, and therefore the initial determinant of features such as the anterior-posterior axis is dependent on maternal genes only rather than those of the zygote. Two of these maternal mRNA, bicoid and nanos, are transported to different regions of the developing egg. Bicoid and nanos are morphogens, which means that a non-uniform distribution or concentration gradient of the protein products of these two genes govern tissue development patterns. These transcripts are involved in determining the anterior-posterior axis of the fruit fly. So through the action of two different motor proteins, bicoid mRNA ends up anchored to the end of the egg closest to the nurse cells, and nanos anchors at the opposite end. Two genes are regulated by the proteins produced from bicoid nanos mRNA, and they're called hunchback and caudal. Bicoid is a transcription factor for hunchback and a translation inhibitor of caudal. So where there is a high level of bicoid protein, we will end up with increased hunchback expression and decreased caudal expression. While nanos is a transcription factor for caudal and a translation inhibitor for hunchback. So it will increase caudal expression and suppress hunchback expression. Now, initially, hunchback and caudal mRNA are evenly distributed throughout the cell. However, after fertilization, bicoid mRNA move completely to the anterior end of the newly formed zygote, and bicoid and nanos mRNA are translated into proteins. Due to the location of these mRNA at opposite ends of the cell, we end up with bicoid proteins at a high concentration at the anterior end and nanos proteins built up at the posterior end. And based on their effects, what we end up with is a high concentration of hunchback proteins, where there is an increased amount of bicoid and an increased amount of caudal proteins, where there is an increased amount of nanos. Now, hunchback is an activator of genes involved in the development of anterior structures. whereas caudal is an activator of genes involved in the development of posterior structures. And so we end up with the development of the posterior anterior axis based on the initial concentration gradients of these maternal gene products, bicoid and nanos. Dorsal ventral axis development in Drosophila is also dependent on maternally expressed genes. Dorsal is a maternal gene involved in determining this axis. In dorsal wild types, the dorsal protein localizes in the nuclei of cells at the ventral surface of the zygote and is a transcription factor involved in the production of ventral structures. In dorsal mutants, dorsal protein is not found in the nuclei of ventral blastomeres, and ventral structures will not develop. The unifying developmental factor in the polarity of Drosophila anterior, posterior, and dorsal ventral axes is that bicoid, nanos, and dorsal gene products all originate solely from the maternal genome rather than from the zygote. Now, homeotic genes are genes that regulate the development of anatomical features, and mutations in these genes result in normal body parts developing in inappropriate places, as can be seen in the pictures on this slide, where we have a wild type Drosophila on the left and a mutant with an extra set of wings on the right. Now, Hox genes are a subset of homeotic genes. They have a conserved sequence coding for a 60 amino acid DNA binding domain, which is called a homeo domain, aka homeobox. The sequence of homeotic genes such as Hox are highly conserved from cnidarians to fruit flies to vertebrates, and Hox genes affect other genes that control organogenesis. And the fact that homeotic genes like this are so conserved suggests that these genes appeared early in animal evolutionary history. So today we developed a better understanding of the role that cleavage plays in embryonic development, including looking at a cell lineage experiment. We developed a better understanding of the role of cell determination through examining how cell transplantation experiments work, and we explored pattern formation in the fruit fly by looking at anterior, posterior, and dorsal ventral axes and the genes involved in the formation of these patterns.